Hi folks, today we're going to learn about mitosis and meiosis, which are two different types of cell division that occur in our bodies. The purpose of mitosis in our bodies is for growth and repair. So as we are growing, or anytime that we damage any of our cells and we need to make new ones, we go through the process of mitosis to get those new cells. When mitosis happens, we take a single cell and double it, basically. We end up with two new identical cells that are exactly like each other. These two cells have full sets of DNA, and we call them diploid cells. The steps to mitosis are prophase, metaphase, anaphase, and telophase. So as we look at this animation here, we see a cell that is going through interphase. It's carrying out its normal daily functions, it's growing a little bit, and right towards the end of interphase, it doubles the amount of DNA it has. So by the end of interphase, it'll have two whole molecules of DNA inside of it. After interphase, the cell will start the process of mitosis. Mitosis starts with prophase. In prophase, you see the nuclear membrane is breaking down, and the DNA no longer is squiggly little lines of DNA, chromatin, remember? It's condensed into chromosomes. Once prophase is over with, the chromosomes will line up in the middle of the cell. That's called metaphase. Next, we go to anaphase, where they split apart from each other. And lastly, telophase, where the two nuclear membranes build back up, and the DNA goes back into the form of chromatin. Lastly, once mitosis is all finished, cytokinesis will happen. It'll split the cell in two. And now we have two separate, identical cells that have full sets of DNA. Now, the whole point of meiosis is to create new cells for reproduction. These cells are commonly referred to as sex cells. You might have heard in humans, they're called sperm and egg cells. Each time that meiosis happens, we get four new cells that each have a half of a set of DNA. When cells have half sets of DNA, we call them haploids. Each of these cells is completely different from each other. They hold the same type of information, but there's different information in each one, and that's why two parents can make different siblings that don't look exactly the same. Once two sex cells come together, since they each have half of a set of DNA, they can start to form a new organism by combining their DNA, giving the organism a full set of DNA. The steps to meiosis are similar to mitosis, except we do everything twice. So we go through prophase 1, metaphase 1, anaphase 1, telophase 1. We go through cytokinesis again, skip interphase, and we go to prophase 2, metaphase 2, anaphase 2, telophase 2, and then we hit cytokinesis again. So here we see another cell in interphase. Same thing as before, it's growing, performing normal functions, and eventually it doubles its DNA. Once it's doubled its DNA, it's ready to start meiosis. The first step of meiosis is prophase 1. In prophase 1, the nuclear membrane breaks down, the DNA condenses into chromosomes, and similar chromosomes, we call them homologous chromosomes, pair up together. Once those homologous chromosomes are together, they can swap around some of their information. This is called crossover. It only ever happens in prophase 1. This genetic crossover allows for a certain amount of variation in the new cells that are being formed. In other words, this is what causes the four cells to be different from each other instead of being exactly the same. After prophase 1 is over, we go into metaphase 1, where the homologous chromosomes line up double file in the middle of the cell. And then we go to anaphase 1, where those homologous chromosomes split up. Next, we go to telophase 1, where the chromosomes turn back into chromatin and the nuclear membranes build back up. And then we go to cytokinesis, which splits the cell into two. Now we have two cells, each with a full set of DNA, but they're not identical. We skip interphase, because if we go through interphase, we would double the DNA, and we don't want to double it this time. We want to take our full set of DNA and divide it by 2, so we end up with a half of a set of DNA. So after cytokinesis and the whole meiosis 1 process, we go straight into prophase 2. In prophase 2, the same thing happens as normal. The nuclear membranes break down, the DNA turns back into chromosomes, but this time there's no homologous chromosomes to be next to each other because they're in separate cells now. So we don't get any genetic crossover whatsoever. We can go straight on to metaphase 2 from here, where the chromosomes line up single file this time in the middle of the cell. And then we go to anaphase 2, where those X-shaped chromosomes get split up into their stick-shaped chromatids. Again, that's the pieces of a chromosome. And now we have half of a chromosome on either side, basically. And then we go through telophase 2, where the nuclear membranes build back up, and the chromosomes turn back into chromatin. And now we can split that cell straight down the middle, and we end up with now, since both of those cells split up into 2, 2 times 2 is 4. So we have 4 cells that are haploid cells. They have a half a set of DNA, and they're each completely different from each other. The meiosis process is extremely important for many organisms in the world. The ability to make sex cells that are not completely identical to each other allows for a variation in the traits of the offspring that two parents can make. For a species' survival, this is really important. Well, thanks for watching through the video. I hope that you got a really good review of the steps of mitosis today, and you were able to better understand the steps of meiosis.